and maybe Abby and Marcus, and then we can go around and I know some of you are already in the interfaces program and some of you are interested. So I'm Andrew McCulloch. I'll be the sort of instructor of record for this course, which I teach with Professor Amaro from Chemistry and Biochemistry, and Professor Terry Sinowski and Tom Bartol from the Salk Institute, as well as Robert Konechny from Chemistry and Biochemistry and Gary Huber. They both work in the McCammon lab. Ariel is in my lab, and Alan is in Padmini Rangamani's lab, and Sachin is in my lab. And then this year, Marcus Hock, who's here, will be our TA. Last year, Abby Teitken was our TA. And we will probably, things actually worked remarkably well remotely last year. So while we will have an in-person venue, it may be just as effective, if not more effective, to do it remotely. They're actually having security problems with running different software and things in the lab. So we will actually make sure things are working remotely first and worry about the physical setup later, because students are spread out all over campus, so it's not very convenient for you to come to the bioengineering building. So we'll try and get it set up so that so that it will work remotely. So Abby, maybe you just want to comment. What was there any downside last year to doing this course remotely?
No, honestly, I think it worked a lot better doing everything remotely, and it's more convenient for people to be able to do homework that way too. So
it's paradoxical for what is sort of technically a lab course, but because it's a computer lab course, you can actually sort of see what's going on your screens, particularly if you have two screens, better, and you can work yourself and follow along the instructor more easily, with your own laptop, and particularly with a second screen. And actually, if anyone doesn't have a second screen and need one, we can just buy you one. We did that last year, and I don't think we even bothered to take them back. We just sent them out, and it would have cost more to ship them back than. So if anyone is in a situation where you need two screens, but you can't get one, then let us know, and we'll just ship one out to you. I should say that that this is a course that's designed for the multi-scale biology specialization, or what we know as the Interfaces Graduate Training Program. And this is technically Lab 2 in that program. And so we set up the enrollments, so it requires approval of instructor, so that we can give priority to PhD students who are either in the program or interested in joining the program. So if you're a first-year PhD student, you've been put on hold for joining because of requiring instructor approval. If you're interested in participating in the Interfaces Graduate Program, we'll clear you right away. And then if you're a master's student or another student, we can see how many students we have, but because we won't be constrained by the space and can do it by Zoom, we should be able to accommodate you. We just want to make sure we give preference to the students who are required to do this course for the Interfaces program. And those students who aren't in the program yet, who are first-year students who would like to join the program, you'd get credit for taking this course, of course, once you're admitted to the program. So essentially, for the purposes of taking this course, you would be an Interfaces student. So can I just ask if there's anyone who is in that category, a first-year student who is interested in joining the program? Well, for anyone who may watch the video, if you are a first-year student, if you're a first-year student interested in from one of the 11 participating graduate programs, interested in joining the Interfaces program, we encourage you to take this course, because you'll get credit for it, and it will actually improve your chances of being admitted with support to the program when we review those applications in the spring. We've been joined now by a few of the other instructors. So you met Marcus, who's going to be our TA, and Abby here, she was our TA last year, and she'll be helping Marcus We have Dr. Gary Hubert here from chemistry and biochemistry. He'll be teaching Brownian dynamics. And I saw Alan Leung from Dr. Padmini Rangamani's lab, and I think this year he'll be teaching a tool that many labs use called Virtual Cell. So what I'll do is I'll just say a couple of words about the course in general, and then I'll just hand over to Gary and anyone else, Abby or anyone else who wants to say anything about the course or answer your questions. So, I also wanted to introduce Kelly Richmond, who's the new program coordinator for the Interfaces program. And we have Irene Hom here, who's our business manager. Kelly's taking over from Mariella, who most of you know. So let me just tell you a little bit about this course. So it's a lab course in the sense that it's hands-on and it doesn't have prerequisites because it's designed for students from all the 11 different PhD programs that participate in the multi-scale biology specialization, which includes biological sciences and biomedical sciences, neurosciences, physics, bioengineering, mechanical and aerospace engineering, nanoengineering, chemistry and biochemistry, and others that I've probably neglected to mention. Mostly we have students from bioengineering, chemistry and biochemistry, and neurosciences attending the class, but we've had a full range. Roughly speaking, the motivation for the program, the whole program is in multi-scale biology, and the motivation for the program is that biomedical sciences has, particularly in the 20th century, became increasingly reductionist. So eventually we knew the genes and molecules, the cells, genes and molecules and genes that make up the systems that form tissues, organs, and living organisms. And increasingly the challenge is to understand how those molecular and cellular scale structures and functions and phenotypes integrate to give rise to the physiological function of living tissues, organisms, organs and organisms. And this sort of theme of integration is particularly well suited to computation. And in fact, computational biology, the different methods of computational biology are all in one way or another primarily integrative. So for example, bioinformatics is essentially the task of information integration, organizing information, sequences, phenotypes into databases and into relationships based on annotation so that we can start to build interaction networks. So I call that information integration. Then as we start to understand the function of components or subsystems within these networks, we can build functional models. So these take the functions of components, put them into a network, and predict the function of the subsystem or the whole system. So here, for example, is a subsystem representing the calcium handling and ion channels in a cardiac myocyte that contribute to the formation of the action potential. And you can think of lots of these kind of subsystems within a cell that with more and more annotated and interaction data will allow us to build more detailed models of metabolism, signaling, gene expression, growth, and electrophysiology, mechanics, and all kinds of transport and all kinds of other physiological processes. But the nature of these models is that they take function in and they put function out. They are systems models. They represent functionally integrated models. They don't represent any of the physical structures. They don't take into account the structure of the molecules or the structure of the cell. They treat the functional interactions. So the third type, and this type of systems biology approach is very powerful, but it's primarily data limited. So if you don't know the interactions, you can't, or the function of the components, you can't model the functional interactions. And when you do, you can. However, there is a way that you could find what the function was by making use of knowledge of the structure. So if we have molecular structure, subcellular structure, cellular structure, tissue, organ, whole body, and even population structures, then we can use more physically based, structurally driven modeling to predict function from structure. And this course is really about the tools that are used at different scales to do this type of modeling, which collectively is now known, at least in the NIH parlance, as multi-scale modeling. I might have been responsible for them using that name. But you'll learn about molecular modeling using tools like the adaptive Poisson, Boltzmann solver, and BrownDye. You'll learn about subcellular modeling using tools developed at the Salk Institute, like MCell, where they model synapses and organelles and transport processes and molecular motions at these subcellular scales. You'll learn about whole cell modeling using virtual cell, and virtual cell also brings in these networks. So this is the point where the systems biology tends to meet the multi-scale modeling. So in a virtual cell model, you can model something here. It's actually the diffusion of cyclic AMP from one end of a cell to another, and some measurements we made in my lab that we then modeled with virtual cell. But in that model is actually a network of cyclic AMP protein kinase A signaling. And then virtual cell makes some of those components mobile through diffusion, and so solves the reaction diffusion equations. And the reactions really come from this systems biology. Then we can take those cellular dynamics and incorporate them into tissues. So this, at the cell level, we may be talking about signaling or metabolism or electrophysiology or mechanical force generation. These events can propagate from one cell to the next, either through gap junctions or through physical interactions, such as the propagation of the action potential in 3 dimensions in the tissue, and ultimately through the whole organ. And then in the case, say, of that action potential propagation, give rise to electrical signals on the surface of the heart that we measure as, say, an electrocardiogram. So we will talk about the numerical methods that we've developed for these type of tissue to organ scale, multi-scale modeling as well. These are continuum methods using finite elements. So these are the different types of tools that we use, and we do this all by hands-on examples using BrownDye, APBS, MCell, all local UCSD products, Continuity, another local UCSD or La Jolla product, and then Virtual Cell, which is from a national resource in Connecticut. So that's kind of our big picture here. And I just wanted to stop there to give you a chance to ask questions, or Gary or Marcus or Abby or Alan a chance to sort of add their own thoughts.
Hello, I had a question. Can everyone hear me?
Yep.
Okay, for this course, I was wondering, well, I guess, I was wondering what kind of software and computer expertise we should have.
So the software we will provide you with, there'll be resources for downloading them. Traditionally, this is the hardest part of the course, and so paradoxically, so we were kind of worried last year that it would become really difficult with COVID, but instead, by using the UCSD Data Hub and stuff, I think we actually might have almost made it easier, thanks to a lot of hard work by Abby. So by using the Data Hub and some environments that UCSD have, it's actually easier to distribute and share the different codes that we use. And none of them are codes or software that you would have been expected to have seen before. And the lectures, they'll include some of the theory, some of the background of how they work, just so you understand what you're doing. But the course isn't about knowing the theory behind molecular dynamics or Brownian dynamics or finite elements or signaling or modeling or reaction diffusion systems. It's more about, you know, the practical problem solving, how you actually do it. And so the instructors, the reason there's so many instructors is because we want to put in front of you the people who are most expert at using these tools. And that certainly isn't me. What I've had the luxury and pleasure of being able to do since we started teaching this course in, I think, 2006, is to be looking for ways, by collaborating with people like Dr. Huber and Amaro and Sanowski, on actually bridging these scales. So you learn the individual tools, but then there's a project, which is really for the final exam. And basically there's a few homeworks along the way just as sort of practice exercises, but the main goal is to do a team project where you choose a problem of your own choosing that involves multiple scales and multiple tools. And the usual rule of thumb we use is you should use at least as many tools as you have members of your team. And so it's not unusual for our teams to have 4 students. And we could give you examples of the presentations they've done in the past. And to do a project where they span from a molecular level model to a subcellular model to a cellular model to a tissue or even organ scale model using 4 different tools in this class. And because you're PhD students, and many of you will be second or even third year PhD students who have, already established interests, it's great if you bring along a problem you're already working on in the lab and work together on it. So this has been the idea behind the course, and I can tell you that some of the projects have basically set new standards. In other words, they've done things that have actually never been done before. Now, they didn't do them to publication level, but in some cases, they turned out to be publications, and in some cases, even turned out to be entire PhDs. So I had a student in this class a few years ago whose basically entire PhD grew out of her project. And that's because she was working with me and Dr. Bartol and Dr. McCammon and Dr. Marrow, and got a lot of help from Dr. Huber and connecting us. So the goal of this course is to really facilitate your ability to do research, but to assume you either know the theory or that you have a collaborator that does. Like, there aren't too many people who know everything from computational chemistry to computational mechanics and all the physiology or biology that goes with it. But that's the nature of modern science. We have to work together. So part of the idea of the teams is that we encourage you to have your teams be interdisciplinary. So, hopefully we'll have students like you from chemistry and biochemistry, from bioengineering, or from nanoengineering, or from biomedical sciences, or from neurosciences. And the more interdisciplinary your team, the more you will get out of this, and the more impressive the experience, or more valuable the experience will be to you. So, so that's the big picture. But the answer is no. You don't have to be an expert programmer. There isn't a lot of programming. Certainly, if you're familiar with using tools, using Linux and things like that, it'll help you. But we usually have at least one person in the group who's good in the lab, and the project groups who's kind of good at that stuff, and then others who maybe know more about the structural biology or the biophysics or some other aspect, or the biochemistry of the problem. Gary, did you want to say anything?
Well, I don't have a whole lot to add. I mean, I've got a few. I can always show a few pictures or talk a little bit about my end of things. I'll be teaching for about a week on what's called Brownian dynamics, and again, the whole idea of this course is about multi-scale, multi-scale modeling. And one of the things that, for an example, Brownian dynamics, this is more down at the atomic and molecular level, and at the most basic level is something called molecular dynamics. And of course, at the beginning of the course, I know I think either Andy McCammon or Romy Amaro will be talking about that. And this is where you're doing a simulation, you're including all of the atoms. Okay, so you've got all the water molecules. If you're dealing with a big protein molecule, you have all the protein atoms, and you have all this stuff in a big box, and it's a lot and you can get some really useful information out of it. And but it takes a lot of resources to simulate every single atom, and of course, the bigger the system, the harder it is, especially if you're hoping to look at stuff on a more cellular level. And so what I'll be talking about in my own particular part, of course, again, you'll learn about the atom level stuff where every atom is included. And then the next level, we're moving more to the molecular level, and we do something called Brownian dynamics, where you come up with a simplified model. And the simplified model is you're no longer including all of the water molecules, and you're including a very simplified version of, say, the protein molecule or other things. And because the models are simplified, you can simulate for a larger period of time, and you can simulate larger things, but of course you sacrifice some accuracy. And so, but again, this is one of these things that bridges the gap. And let me see, let me share a screen. I'll just show you a quick little animation, nothing really too spectacular. Let me see if I can share the screen here.
I think you should be able to.
Okay, yeah, hold on. Here we go. I've just got a simple, let me find where I am here. Yeah, this is just what I would call a Brownian dynamics simulation of a small protein molecule. And let's see. And what this is a simplified version of a protein molecule. And what it does, it doesn't include all of the water molecules, and it also actually doesn't even show all of the atoms, but this is what's called an intrinsically disordered protein, which is a protein that doesn't really fold up into a fixed state. And that's one of the research areas that we're interested in with this tool. And so with the software that I'll be teaching you, I'm the main author on it, and you'll get to generate simulations like this and use them in your projects. And of course, if you run this further, it'll change shape. But this is an example of again, one of these.
I don't think we can see it. I don't think we can see it, Gary.
Oh, you can't?
No.
Huh, okay, let me try.
I thought it was here for a while, but.
Can you see my screen, or
you
can see me, I guess. Let me see if I can try sharing again. Let me see. I'm not exactly the Zoom meister here.
So you have to have whatever you're displaying opened, and then you just click on that one.
I thought that's what I did. Okay. Oh, hi, Irene. Long time no see.
I was gonna say hello. How are you?
Yeah. Hold on. Okay. Let me do this again here. Nope, don't want to do that. Anyway, I won't take up too much time with this if I'm not smart enough to share my screen. But let's see. Okay, hold on. Oh, heck. Here we go. Okay, can you see anything?
No.
Okay, let's see.
Is it a slide or is it a?
It's like an animation in a window.
Oh, I see.
Maybe that's okay. Well, anyway, okay. I'm not going to spend a whole lot of time on that, but basically, what I'll be doing, I'll be teaching you in my portion of the course, and of course, there's lots of other softwares. I'll be teaching you about this particular software called BrownDye, which does these molecular simulations, and it allows you to do larger things than when you include all of the atoms. And I'm the one that wrote the code, so I'll be able to help you a lot with it. And again, there are some people that have gone on and used this in their own research projects. And also, as far as the computer skills needed, my software does not have a very fancy user interface, and so you probably will have to learn how to manipulate files and things like that, and know some of the commands in Linux, which some of you probably do and some of you may not, and that's fine. you'll pick it up real quick. And again, the TAs will be very helpful. I remember last year when Abby was the TA, she was very helpful with helping people learn how to manipulate files and switch directories and do that sort of thing. And this sort of thing is going to be important when you do bioinformatics, you know, if you continue on with this sort of thing. And so so at least with my part of the course, because it's mostly command line driven, where you pop up a terminal and you type commands, kind of the old school way. You'll definitely get a good workout with that, and but you'll be able to get some nice pictures too if you feed it into some of the animation software. You can get some really cool things. Like I said, for some reason, let me try sharing the screen again, see what happens. Desktop one. Oh wait, VMD. This might be it. Okay. Allow Zoom to share your screen. Okay. I guess it's not happening. Well, okay. Well, anyway, I guess I should have logged in and done a trial run. Oh, here we go. Oh, I know it is. There's some sort of a security thing that's keeping me from doing it. That's strange. That's never happened before, but you never know. Well, here we go. Allow Zoom. Okay, check that box. Okay. Now let me try it. And again, I will not spend too much time on this. Desktop one. Here we go. Okay, there we go. Yes, this is an example of a, can you see it?
Yes.
Oh, good. Okay, cool. Yeah, this is an example again of a simulation of a disordered protein molecule. And you notice it doesn't have all the full details. You just see the backbone, and then you see simplified versions of the side chains. It's a very simplified model, but it's useful because it can study these particular types of molecules. And this is an intrinsically disordered protein. And this is the sort of thing that's becoming more and more apparent. You know how most proteins, they fold up and form a particular shape to do their functional. These things actually stay disordered, but they actually do useful things. And they're finding out more and more about these things in cell biology. So this is just an example. This thing started out as a helix. Now it's kind of folding over into a hairpin. So if I run it more, you'll see it change shape. So anyway, this is something that was generated from BrownDye, again, using a Brownian dynamic simulations. This would have taken a lot longer to do if you had included all the full-blown atomic details. And then there's other, let me see, and I'll show you some other cool pictures on the browser. Okay, hold on. Let's see. We share something else. Share screen. Browser. Here we go. Okay. And okay, so this is the website of BrownDye. And one of the things that we also study are two proteins associating with each other. And here's an example of two proteins. These squiggly yellow lines show trajectories, you know, where the thing actually travels, where one protein travels in order to interact with this other protein. This is just a still picture. And then here's a few other pictures that were actually generated using the software. One of the things we're interested in is finding rate constants of two things binding with each other. And that's a very important thing, protein-protein associations, for example. And here's some images that were generated by people that used our software. And here's a very brief movie. I don't know, you probably can't see too much, but there's a molecule that's binding to one of the components of I think an influenza virus, I believe. That's a sialic acid, which interacts with some of these viral proteins, and you can see it kind of dancing around, and then it binds to its binding site. So this is just a real quick flavor of the kind of stuff that you'll be learning about on the molecular end of things. And of course, then this also goes up to the next level, which is more the tissue level, which of course Dr. McCulloch, his software specializes in. And so people have done joint projects where they've used the molecular dynamics and the Brownian dynamics to get some numbers that then can feed into some of the tissue level models, and that feed into that. And so you'll get a taste of this. If you take this course, you'll get a taste of that. And again, you might end up using both a software at the molecular level and software at the tissue or the systems level in your final project. And again, it might go on to a publishable project, or it might just be a good learning experience. It all depends, but either way, you'll learn something about it. And then also, I know, I don't think Robert's here, but he's going to be one of the instructors. He's going to teach you how to use a piece of software that calculates the electrical field around these molecules. And that's a big deal, because if you're going to simulate these molecules, you need to know what the electric field around it looks like, okay, because that's how they interact with each other. So Robert Konechny will teach you about that software. And again, he was one of the key people that helped develop that piece of software. So you'll be hearing from the experts on a lot of this stuff. And then I think, and then of course there's all sorts of other cool packages that you're going to learn from. And I know Dr. McCulloch has the big picture. So I didn't really have any formal presentation plan for this. Of course, you'll hear more of it when you take the course, but I don't know if anybody has any questions about any of the simulations or about more general stuff.
Thanks, Gary. So you're starting to see the modules. So first we'll have molecular dynamics with Dr. Morrow, then we'll have the molecular electrostatics with Dr. Konechny, and then by using those molecular dynamics trajectories and the electric fields that get computed with the Poisson-Boltzmann solver, then you can start to use the Brownian dynamics to do longer timescale solutions and protein-protein interaction solutions using BrownDye. And then you can keep going. And, over 20 years ago when Gary and I met and Dr. Bartol and I met, these were just a vision of what we hope to be able to do in the future. And then as we've taught this course, we've started to find new ways to bridge the scales. And now, Abby and her research actually is basically spanning all of the scales, starting with molecular dynamics models of the effect of a small molecule and going all the way through to its ultimate effect on the function of the whole heart. And Marcus is also he uses BrownDye and he uses APBS, and so Marcus will be a really helpful TA. And then after Gary, the next group that we hear from is the group from Salk that have developed the MCell tool for subcellular Monte Carlo modeling, and Tom Bartol is here and he can tell us about this. And I'm not sure whether you have any pictures to share or anything, Tom, but you usually do.
Sure, absolutely. Yeah, like Gary, I don't have a formal presentation, but I just want to meet you all and tell you a little bit about what you're going to be learning in the class. Hopefully you'll be taking the class. Me and my group over many years have been developing software called MCell, a Monte Carlo cell. It's a simulator of reaction diffusion of biomolecules in realistic 3D geometries, subcellular and cellular geometries. And because it's all three-dimensional and all realistic geometry, a big boost for us was several years ago, the open source software community, computer graphics modeling community has invented and came out with an open source software called Blender. And Blender is software that lets you make, Hollywood animated movies, but it also lets you make realistic models of cells and look at 3D, create and curate and work with very realistic three-dimensional models and render beautiful pictures of them. But the software, the Blender software has a customizable user interface that you customize in Python. And what we've been able to do was layer on top of Blender a set of user interfaces and control panels that let you create entire simulations of the biochemistry and diffusion and interactions of molecules. And basically take the MCell physics engine, which simulates individual diffusing molecules and their collisions with each other. Make that physics engine be one of the supported physics engines that Blender knows about. So in Blender, you can do Newtonian mechanics and have automobiles crashing into each other and walls falling down and things like this. But you can also have a Newtonian physics engine called the bullet physics engine, which is kind of built into Blender. But we were able to make MCell be one of the particle physics engines of Blender. So it's kind of cool. And recently we have a recently released new version of MCell called MCell version 4. What's new in MCell version 4 is you can create your model as a Python program. And there's a whole Python API for interacting with all the components of a very powerful Python API for creating and querying and introspecting and interacting with all the different parts of the model that you've built. And because it's all done in Python, this means you can couple to other scales in a much more readily immediate way than we could in the past. So this is the first year in the course where we'll be introducing to you MCell version 4, and I'm very excited to see what some of you guys might do with your multi-scale modeling projects and coupling MCell to the other scales. Mm-hmm. Previously, MCell, the way you interacted with MCell was to create your model using a language called the MCell Model Description Language, which is a domain-specific language that is very nice and convenient, but not very extensible and not readily able to be coupled to other physics engines. So now with this Python interface, it's kind of the whole universe opens up, I think. So, but now let me share my screen for 1 second here, and I'll show you. Let's see, I've got some other things going on here on my screen in the background here, but let's see. HD. Yeah, okay. I have a version of the movie with sound. I'll probably turn the sound off. Sound? Yeah, I'll just do it without the sound. And here's an example of a model. If you take the course, I'll go into great detail about this model. But this is a model that shows what can be done with MCell. This is a model of the release of neurotransmitter from an axon in response to an action potential traveling in the axon. And then the postsynaptic response of the AMPA receptors and glutamate and NMDA receptors on the postsynaptic cell. We're gonna, in the simulation, I'll explain as we go along, but the simulation spans many many different time scales. So we have 1, 2, 3, 4 decades of exponential time, or time on a log scale, starting at a few microseconds per time step, going up to 100 milliseconds at the end. And so I'm going to let me move this out of the way so it's not in my face. And see, I can probably go to full screen mode here too, right? Full screen mode, there we go. And I'll hit play. And we're going to be zooming in on this, but what you're looking at here, the yellow is a dendrite, the green is an axon, and the blue is astrocyte. And this is a 3D reconstruction from a rat hippocampus area CA1. This is a Schaefer collateral synapse. The purple thing here is a mitochondrion inside one of the synaptic boutons. As we zoom in, you can see the little particles diffusing around. Those are free calcium ions in the cytoplasm of the axon. And bright green color indicates that an active potential is arriving and the voltage-dependent calcium channels are opening here. And calcium diffuses in through those, diffuses through the bouton and reaches into the active zone where the little red particles are snare complexes and they bind calcium. And when they bound enough calcium, they release neurotransmitter. Right there was a release of about 2,000 molecules of glutamate into the extracellular space. And they cross the extracellular space and bind to the green and blue receptors here on the membrane. Though the green are AMP receptors and the blue are NMDA receptors. And when NMDA receptors flicker open, they let in a little burst of calcium that you see come in. And the calcium can bind to calmodulin, which are the little purple, they go by fast, are hard to see. And then the activated calmodulin can bind to CAM kinase 2, which are the bigger cubes here. And there's going to be a second release, a neurotransmitter that occurs asynchronously in a moment. But the bright yellow color of the dendrite indicates that a back-propagating action potential has occurred in the dendrite, and that depolarizes the membrane and allows the NMDA receptors to let in even more calcium. And now that the calcium builds up in the head of the dendritic spine and activates even more calmodulin and kinase 2, which eventually, over a period of many seconds or minutes, can lead to potentiation of the synaptic efficacy, a process called long-term potentiation. But that's not modeled here on this time scale. So each, what you're looking at here is, you saw diffusion happening, you saw interactions of molecules happening and molecules changing state and being released and diffusing in the extracellular space. And then, the red particles here are the glutamate transporters that can reuptake the glutamate from the extracellular space and absorb it back into the cytoplasm of the astrocyte. These little particles here on the membrane are the PMCA pumps that after the calcium enters, the PMCA pumps can extrude the calcium back out into the extracellular space to try to maintain calcium homeostasis. This up here is the endoplasmic reticulum, and the little green particles on that are the circa pumps. So we have all these different molecules, and they all behave according to a chemical kinetic scheme with rate constants for binding, unbinding, opening, closing, translocating, transporting, all kinds of enzymatic functions that they can have. So if you know this, so this is at a length and time scale, beyond BrownDye, and in BrownDye you're trying to calculate many different things. One of the things you're trying to calculate is what is the rate constant for binding? What are good values for that? And in MCell, we're saying, what if we already know what the rate constants are? We can leapfrog those timescales, the molecular dynamics and Brownian dynamics timescales. We can leapfrog those up to this timescale where the typical time step is a microsecond rather than femtoseconds or picoseconds or nanoseconds. We're at the microsecond timescale per time step. And the questions you can ask are, what happens at this subcellular or more macroscopic scale? If I put together these molecules and if I can put them in space, in their spatially organized anatomical domains that we know that we know they exist in, what is the behavior of the system like that? So these are the kinds of questions you can approach with MCell.
Thank you, Tom. Any questions for Dr. Bartol? So I think you can see that this is a fun course with a lot of sort of high-powered tools that you'll get to learn and use and so on. And Tom, before you came, we were just pointing out that last year seemed to actually be possibly even more successful using Zoom. So even though we will have an in-person setting, we'll probably do this class remotely just because it's actually better.
Yeah, and I can amplify that. I agree with that 100% as well. And when in March of 2020, right as the pandemic was beginning, we had booked a venue to hold the next MCell and cell modeling workshop where we were going to teach MCell and other tools to students. And it was going to be an in-person workshop, as it always has been. It was going to be held here in La Jolla on the beach, La Jolla Shores. It was going to be great, right? But we had to cancel it, and we said, well, should we cancel it altogether, or maybe we can try to do it, as a virtual workshop? And we put our heads together, and we delayed it by a month and got reorganized and announced it. And instead of having 25 people in person, as we would normally, that would be the limit that we could handle in person because of the venue, we had 95 people from all around the world sign up for this workshop. And it was the most successful, rewarding thing for all of us involved in it. And so, so this past summer, again, we had the virtual workshop again, and we had 100 people, come, some repeat students, but mostly new students. We had people who could never have come to an in-person one. We had one young man from Nigeria. We had lots of students from India. We had students from Australia, and they were attending at all hours of the day and night, right? In their time zones, right? And we all, the whole crew who put this on, we said, would we ever do an in-person one ever again? Because the virtual one is so great, and probably we might have some in-person ones, just because. But I think we'll always have the virtual one going forward.
I think for this course, we used to have to spend a lot of time to account for all the changes in the lab that had happened and get the latest versions of the software installed. And so we would spend a lot of time to get the environment set up for you in the physical teaching lab. Now in this course, we'll spend a little more time. You will have to spend a little more time getting the environment set up for yourself on your own computer. But it turns out not only is that much easier these days with various virtual machines and other environments that we can use and the data hub at UCSD, but also it makes a course much more useful to you, because it's one thing to say, well, I could go over to one room and run some software, but if you can do it yourself, anywhere, on your laptop or connecting to a server, that actually makes everything you learn more practical and more translatable. So, you may find that a certain amount of the class is spent on logistics of actually installing software and learning some of these environments, but it will pay off for sure, because not only, even if you didn't use any of the tools in the class, and chances are you will, but even if you don't, just learning some of those techniques and how to take advantage of some of those virtual machines and data hub resources, you will almost certainly find useful. So at that point, the next point we would go to is to learn virtual cell, which is sort of a whole cell reaction diffusion modeling system. So now we're modeling similar processes to what you would see in M-cell, but at a continuum level. So just modeling distributions of concentration rather than following individual particles. And the reactions are driven by the sort of signaling networks that are and reaction networks that we will take a short break on, and that Ariel and I will teach you about. And then Alan will come along and talk about a virtual cell. Alan, was there anything you wanted to say?
Yeah, just gonna say something similar. You know, I work on similar things to Tom Bartel, but at a higher level of like abstraction. So not so concerned with the individual movement of molecules, but interested in longer time scale behavior of neurons and calcium signal transduction. And I think one of the greatest things about VCell is how accessible it is to someone who's, you know, not quite as interested in like incorporating modeling into their ordinary workflow, but wants to see some of the powerful things about modeling. So it's, you know, very user-friendly, very easy to learn, and it's something that you could probably quite easily use in your real life as well.
One thing, Virtual Cell is the only tool in this class that isn't a La Jolla developed product, but it's also, you can use it through your web browser. So it's kind of a relatively easy tool to use. And it's also designed, if you have microscopy images, that you can generate your 2- or 3-dimensional modeling domains from your microscopy images or your stacks of microscopy images. So that's kind of a fun part about Virtual Cell. And then from there, we move on to continuum modeling of mechanics, transport processes, or electrophysiology with continuity. In the past few years, we've focused on electrophysiology because we have a lot of students interested in neuroscience. And I think this year I may actually do mechanics instead because I know that's an interest of Marcus and Abby. And we may also get so far as to talk about how we can make models from patient images. So that's something that we've put quite a lot of effort into in the past few years, particularly using machine learning. So we will, the last couple of weeks of the quarter, we'll do some examples of whole tissue and organ scale continuum modeling using either the mechanics or electrophysiology of the heart or both as examples. Before that happens, though, we'll get you to form your teams, and I'll give you some prompts sort of starting in the second half of the quarter on how to form your team, and I'll ask you to sort of give us a fairly informal feedback of what your thinking as a team is, and we'll give you some bit of guidance based on our experience. So that the last few weeks of the quarter, things will sort of taper off, and we'll probably have maybe the last week there won't even be any classes. It will just be a time for you to work with your group together, and then we'll find a time for the groups to present their projects during finals week. You know, if we only have a class of, say, 16, then that's only 4 groups. And if we each take 15 minutes, it's only an hour. So, so it's not, hopefully it won't be a mad rush at the end of the quarter and a long project presentation, but just a more of a fun team exercise. And I know in the past, Dr. Bartol and Konechny and Huber and members of the Rangamani lab and my lab have always been more than happy to help the student teams, you know, get their codes, their models working. So I think that's about all we have. We're coming up on 4 o'clock. The one disadvantage of doing this in person is we don't get to give you pizza, but if there are no more questions, we'll close it out. And I think we'll have a recording for any of your friends or colleagues who might be interested who missed it. Okay, well, thank you all very much, and we'll see you next quarter.
Thank you.
Thank you, everyone. Bye.
Bye.